Okay, good. Oh yeah, so my talk is called From Foe to Friend, so from enemy to friend. And I'm showing you an evidence for the recruitment of florivores as pollinators, which I found also in Costa Rica in the tropical field station La Gamba, which is run by the University of Vienna, by the way. It's a beautiful place in Golfito, if you want to visit it. And first I have to start with this large pollination system, which is shared by lots of genera that we heard from Tom and Alejandro. So it's the scarab beetle pollination system in the neotropics. It's a pollination system that is not well known, actually, maybe because it's nocturnal or not so many people have studied it, but all these genera and more aroid genera are pollinated by these beetles, and this covers probably about 1,000 species of neotropical plants, which are pollinated by these large dynastines, so rhinoceros beetle relatives, actually. To get into this, I have to start a little bit about this pollination system, because it's a little bit complicated. So, as an example, Dieffenbachia aurantiaca, this was the first plant I was studying several years ago. And they have a female phase, which is much shorter than the one in Anthurium. It's only one night. So in the evening, it starts to heat up. It's producing temperature. It's a thermogenic event up to 6 to 9 or 10 degrees warmer than the ambient air. And you can also see this on an infrared camera always. And thanks to this picture, Santiago, that was your camera. And then a floral scent is emitted. This you can smell like from 20 meters away. You can sniff the philodendron and go there, usually. Some are not so strong, some are even stronger. And this attracts these cyclocephaline beetles, 2 species in this case, and they go into this chamber because it's the rendezvous site. You see nocturnal camera video here where the beetle is, it's totally dark. The beetle is finding it only by scent and walking down, and you already recognize these other insects there crawling around. These are Miridae bugs, and it will get exciting with these Miridae bugs soon. But let's focus on the scarab beetle. So it's going into the chamber where, in the best case, a female is waiting or a male is waiting for the other sex, and then they will copulate. So this is the rendezvous site. These beetles are not finding each other on a twig or anywhere in the canopy or in the ground. They need these plants, they need these flowers to smell and to find their mate. And while they're mating, they also touching the stigmas here, and it gets better. They are surrounded by staminoids, like food bodies, where they can feed on. So they're mating, feeding, pollinating the plant, and you see here this smeary pollen, which they have on their body, brought from another inflorescence, and they touch these stigmas and pollinate it. Floral food bodies are in all these cyclocephaline pollinated species abundant in and diverse. For example, here in Syngonium, schottianum, you see, like in a lot of philodendrons, here in the intermediate zone between the female part with a lot of female flowers and the male part with a lot of male flowers, you have these sterile flowers. You find this also in Xanthosoma, in philodendron. And in Dieffenbachia, it's a little bit different, as you saw. But this is the most important food source for these beetles. And on the next day, 24 hours later, where they had the whole day in this chamber, laying around, waiting for the night because they're nocturnal, and then they crawl out. So they crawl out, and the pollen is produced, so they feed on the pollen, and the spather, so this leaf which forms this pollination chamber, is closing. Slowly but quick, so in 1 or 2 hours it's closed, so they have to leave it again. So party over. And Also, these mirid bugs and several other florivores are known to visit these inflorescences. So they smell strong, they have a lot of pollen, food bodies, they have this chamber heat. So of course they're attractive to a lot of other insects which find them. And especially these mirid bugs are everywhere abundant. Where you go and you see a philodendron or so, you will always also find these mirid bugs and Dieffenbachias. They use it in the same way. They have a rendezvous site here, and they're feeding, but they're usually not feeding on the food bodies or entering the chamber. They're just feeding on the male flowers. Probably they're somehow like macerating the pollen and getting the pollen nutrients. And they are also attracted, we published this several years ago, they're attracted by the same scent as the cyclocephaline beetles, so they're hitchhiking on this communication channel. But they are not pollinating. They're not going to the chamber, they're not carrying pollen. See, they're not covered with this pollen. Why? I will explain soon. So they are florivores. They are enemies, actually, okay? Also in Syngonium schottianum, you can see these bugs around. If you find pictures in the internet of these plants, you will find also these bugs around. So to sum this up, this pollination system, you have the beetle down in the chamber, feeding on the food bodies, no bugs there. You have these pollen grains, which are usually smooth. They are on the body of the beetle. And you have strong floral scents, several of them known, that attracts these beetles and these florivorous plant bugs, usually at night, okay, during the night. So, and I was studying many of them from a PhD thesis in La Gamba, looking at all philodendron, Adelonema, Syngonium, and so on. And suddenly I found this Syngonium hastiferum. I thought, oh, another one, good, let's do the same work as always, climb up, put in the thermocouples, wait for the evening, nothing happens, no scent, no insects, weird species. Next morning, checking it again, I saw, oh, there are a lot of bugs, but no beetles that came during the night or so, and it was smelling during the day. Interestingly, these bugs entered the floral chamber, so they were sitting on the female flowers. They were also feeding on other parts, of course, so they did the same job as always, but they were potential pollinators. So I thought, oh, this is interesting. So I investigated this a little bit more. They're in the morning, entering the floral chamber, and I saw there are no food bodies. This is actually kind of a tail, so like very slim here. And hope this works. Oh, no, wait a second. This should start. Can you press any button or something that it starts? This should, yes, it is, yeah. So this is a time lapse. Oh, can you click on this? Oh yeah, so this is a time lapse. It makes pictures every 5 minutes over 3 days. So you see it's opening during the night and it's starting to produce the scent. All these bugs are coming. They are going inside. Sometimes there is a lizard. That's why they are not always there. They hide inside. Then, I think it's already the next day, they're attracted again. Even more scent. Lizard, no lizard. And then the male phase starts. They really go on the male part. It's also closing, but not completely. And that's it. Then it's just bending down, and they all left. Probably, or we know now, being attracted by other inflorescences in the same area that have just now started the female phase. So during the male phase now, 2 days later, like in the Cyclocephalini pollinated ones, pollen emerges. But in this case, the pollen is kind of powdery. It's not this slimy pollen that you saw on the beetles. It's a little bit like snow. And this beta also drops down. So they have no hiding place, so they have to go. And they're covered with pollen. Others than, if you see here in a Philodendron grandipes, where also the bugs are sitting on the little bit different species, but the same genus of mirid bugs, sitting on the pollen, having no pollen grain on their body, because this pollen is like toothpaste. You need to step on it, or you have to be heavy to get it You see here in Philodendron popinoi, the bug is sitting on these pollen strains, not even covered by any pollen grain, although it's totally surrounded by the pollen. So there's a really important thing, how this pollen is sticky, how much pollen kit it has, how heavy it is. Even here on Syngonium oduberi, feeding, the pollen strains coming out, no pollen on the bug. So, So this is the beetle pollen in a scanning electron microscope picture, covered with pollen kit, surrounded by dead pollen grains, calcium oxalate crystals. Yeah, this is like a microgranulate toothpaste. And the pollen of the Miridae bug, pollinated Syngonium hastiferum, is like snowy, doesn't stick together, and it has little spikes. It's echinate. And I have the same problem like everybody. After a few minutes, it stops to work. I don't know why. Hello. Okay, so you see here the pollen between the hair, although they look kind of smooth, they have little hair, and the pollen is everywhere on these bugs. Each part they clean a little bit their body if they can, and then they leave. Okay, please. Yep. And they touch. This is the most important thing. They touch the stigmas. When they go to a female phase inflorescence, they enter this chamber and they touch the stigmas with the pollen, which is still on their body. Oh, I think it worked again. So also, I always collect the floral scent and analyze it. In this case, I found a compound which was unknown. Nobody could tell me what it is, so we had to do NMR. And found out that it looks like this, which tells you nothing. It is the name, which will also tell you a little bit more, but some kind of alcohol. Actually, you need to synthesize it, and then we call it gambonol. It's a better name. It's covered in La Gamba, so gambonol. And then even better, after La Gamba, we did biotests. So here, a comparison. This is the inflorescence. This is the inflorescence if you put a bag over it, which lets the scent through. And this is now a cone. So, little side story. Another PhD student in Germany dedicated half of his PhD to synthesize this compound. After a while, he stopped because he told me, sorry, the next step includes an explosive reaction, which might be dangerous, so I will not do it. So I was going to Regensburg, eat a Schweinsbraten with him at lunch, and gave this part of this talk and convinced him to please synthesize this compound. Take the risk of your life, man. So he did it, and after a while, he said, oh, I have this compound. I have a few microliters for you. So he was very short because we were going to Costa Rica. So he kind of came to the airport and we met somewhere and brought this compound to La Gamba and put it on the filter paper at the same time when the inflorescences are flowering. And really, it was crazy. A cloud of bugs were arriving and going there. And where's the flower? But they were just staying there for a long time, expecting to find some pollen eventually, but they didn't because we caught them. But yeah, so the biotest worked. And a little bit background now. So the beetle pollination is ancestral it's an ancestral state in the whole clade Caladiae. We heard already from Alejandro that Xanthosoma and Chloris beta are also beetle-pollinated. He confirmed that now with this cucullionid beetles, Caladium as well, and Syngonium and Caladium, and also all the Syngoniums that were known were beetle-pollinated. So there's some new pollination system deriving in this genus. Now it stopped again to work. So now the second part here is now this was like Syngonium schottianum and the other aeroids, and here you have Syngonium hastiferum, which differs in that the plant bugs are in the pollination chamber covered with spiky pollen without pollen kit. There are no food bodies, and they are attracted by a specific compound during the morning hours. Okay, please, next. So we have a pollinator shift here from, interestingly, from a former florivore that have led to changes in a lot of floral characters in the morphology, in the pollen morphology, the scent chemistry. The thermogenesis is different because it's also during the morning, and the timing of antithesis totally changes. And with this, I have to go even more back to the evolution of insect pollination as a whole, because the hypothesis about insect pollination is that herbivores started to eat pollen and ovules, and from herbivores they became florivores, and somehow plants evolved to make them pollinators. So this is kind of a term is called antagonist capture. So the plants made it possible to make from these florivores, from these foes, a friend. And even if another hypothesis is that carnivorous insects that choose ovules and pollen instead of insects started to be florivores, but in the end they turned from florivores to pollinators. And next, please. This is some kind of picture, nice picture, which showed, it's a recent paper, that in the early Cretaceous, florivory was also very common. You had a lot of insects going on and destroying the flowers and becoming pollinators. And you know, fossil records from like great snapshots with pollen on beetles in amber or in the stomach of a fly. But, please, next. But tracing the possible origin of this pollination is not so easy. So it is hard to show this evolutionary process. You know, you can say, okay, it happened like this, probably because we have a lot of puzzle pieces. But here you see an actual shift in a kind of recent example from florivory to pollination. And this is rare to show this. So we have this recent example where a beetle-pollinated plant lineage where florivores are omnipresent made a shift to make this florivore pollinator, and this can drive floral diversification because then more plants can derive from that. And next. So we were able to publish this in a good journal, Current Biology. And next, please. And we, of course, we tried it in Science, but they rejected us. But later, they wrote me, please, can we, sorry, can you go back just shortly? Please, can we write about it? And it turned out to be between COVID and the Neanderthals, which is hard to manage, because these are the topics that were most important, of course. And to see an aeroid there, I was quite proud that we, and of course they said plant turns suspected crop pest into pollinator, which is a little bit okay, but mirid bugs are really known as crop pests worldwide, and they tend to destroy flowers and of a lot of crops, which then cannot produce. So probably they were hoping we find the next attractant for all these crop pests and eliminate them. But, so if you are interested, there is a film, a video abstract on YouTube where you see 5-minute video on this story with lots of more pictures and videos, which I cannot show in this time. And we are now studying more Syngonium species, of course, here in the Botanical Garden of Munich. This is Syngonium schottianum, where we finally got the scent. We need to go to La Selva now and try to track the insects and see what's going on. And I say thank you for your attention. Thank you for a lot of people that helped me, and grants, and for this great symposium. Thank you very much.
All right, thank you. Any question?
Thank you. That was such a wonderful story. I'm really curious about the pollen morphology, because the other floral traits like scent and thermogenesis, there seems to be a lot of precedent in them evolving pretty rapidly, but the pollen morphology evolving as well? Like, do you know how common that is in the Araceae?
I mean, there are some genera where this has been studied, where different shapes of pollen are present. For example, Amorphophallus has a lot of different pollen shapes. I mean, and also in Anthurium, there are a lot of different pollen shapes. Nobody knows why they look like they look like. Maybe it's for some specific insects or not. In philodendron, they all look the same as far as we know so far. But I don't know how common it is to change the pollen to get a new pollinator or like this. But it happens, yeah.
Thank you, Florian, for this very nice story. And well, I have the question. So I assume that the plant will suffer from
the bugs, you mean?
bugs which are pollen feeding. So is there any quantification how much the advantage is to get them rid?
I mean, you mean to get rid of them or to make them a pollinator? I mean, they were not able to get rid of them.
Yeah.
So maybe that was the only way. And I guess if they are numerous, if there are 200 bugs sticking their proboscis into the anthers and somehow macerating the pollen, it might really affect the pollen export or the germination of these pollen grains. But we did not look at some quantification of this, but this would be a nice ongoing, like a nice story to do, yeah, of course. I mean, they destroy also the spathe a little bit, which is not a big deal, but this pollen stuff, yeah, we should look more into it, yeah.
Florian, what about fruit set? Those plants can produce fruits, and the fruits are in the complete female part, or?
Yeah, sorry, I put this slide out, but yeah, if you bag them, you will not have fruits, they abort. And if the bugs are there, usually they produce fruits. And only the female part, like in most aeroids, or most of these aeroids, produce fruits, the top part falls off. Or
But I mean, like for example, in Chlorospata, only the top of the females are able to produce fruit, and the female flowers in the pollen.
No. They, a Syngonium in fructescence it's like a little oval-shaped thing, and usually they all have seed. Usually, I mean, they produce some kind of fleshy stuff, and there are the seeds inside. More or less, depending on how good they were pollinated. It's not really a pattern where you see the bottom, not top. Yes, they smell strange.
One last question.
Oh.
Thanks, great talk, amazing story.
Thanks.
You had mentioned kind of in passing that these myrid bugs, which I'm not familiar with, that they are an actual pest on crops. Have you considered, or is it more maybe the guy who decided to risk his life? Has anyone considered using them as attractants in the context of trying to prevent crop loss?
I mean, no, because I mean this compound, a little bit of it to produce it is quite risky, it seems, and it specifically attracts this Philodendron diffenbachia Syngonium bugs. I doubt that if you use it in North America or in Europe with some kind of cauliflower or cucumber pest, that they will go there. But we could give it a shot, of course, but it would be quite expensive, I think. Then it would be easier to use this jasmone, which attracts the other bugs and the beetles. But this, yeah.
It's nice if you have something that's very specific to one pest species, but I appreciate it.
That's where chemical ecology actually came from. People wanted to see a pheromone of a moth and to attract the pest. So this is actually the main thing of chemical ecology. So if somebody would like to collaborate in this and bring me there to look at the specific pest bug, I would maybe say, yeah, but I didn't try.
Thank Thank you so much.
Thank you very much.